Welcome everyone to our episode 75 of our podcast Cultivating a New Generation. In today's topic of the season 4, we are going to speak about an amazing topic about how our environment, understanding how your environment and our environment regulates your genes. So we are going to um, demystify the concept of epigenetics and how we are not at the expense of our genes or the inheritance, but we are more the owners of how we reprogram ourselves and the signals and our metabolism. Everything that we have inside is a response of the environment. And of course, it has to do also with the beliefs, with the emotions, and with the attitudes that we are constantly repeating in our days. So, in order to understand this topic, I used the article. Cavalli is the first author from 2019, and the title is Advances in Epigenetics Link Genetics to the Environment and Disease. The journal is Nature, and is the one of the most renowned journals that you can look for. And we are going to define what is epigenetics and how was the history of this amazing science. Biologists have long sought to understand how a fertilized egg can form an organism composed of hundreds of specialized cell types, each expressing a defined set of genes. Cellular identity is now accepted to be the result of the expression of specific combinations of genes. This expression pattern must be established and maintain two distinct traits, but connected processes. The pluripotency of the initial cell, that is the availability of the initial cell to give rise to a certain specific type of cell, and the establishment of the cell types depends on a large extent on the coordinated deployment of hundreds of transcription factors, which are helpers in this process of the decodification of the genes that bind to a specific DNA sequence to activate or repress the transcription of cell lineage genes. So what is the easiest way to understand all of this information? Let's just say that this book is in English and I'm going to start promoting it or selling it in Mexico, and I need to translate this book to Spanish. So the people that are going to help me to translate, to review, to edit, and to do all the work to get this book in the Spanish language are the helpers of the translation process. In the DNA expression, we also have factors, cofactors, environment signals, many other players that are going to help to read a sequence of a gene. And these kind of transcription factors are going to bind to a specific sequence in the DNA and are going to help to signal the other factors that are going to read the enzymes that are going to read the information and then give a first translation of the information. So that's what is going to happen. We need this all amount of players so that we can start having a much better quality of proteins. So this establishment phase corresponds most closely to what is generally cited as the first definition of epigenetics, which was given by Conrad Waddington namely the study of the mechanism by which the genotype produces the phenotype in the context of development. So what does this mean? This is the genotype is the information that is in your genes. Let's suppose that this is a sequence, my hand is a sequence of the gene, and this is the genotype. The reading, the translation, the addition of all this information is going to give rise to a phenotype, which is the color of my skin, the color of the eyes, the color of the hair. Everything that we see is the expression, the phenotype. That is how you can understand much better the development of the information. In terms of a protein, it is the shape of the protein, it is the function of the protein, and it is what is going to do in the whole orchestra of the metabolism or whatever it is, the function. The maintenance phase often involves a plethora of non-DNA sequences, specific chromatin factors that set up and maintain chromatin states through cell division and for extended periods of time, sometimes in the absence of the initial transcription factors. So, what is this chromatin and all of this kind of terminology that I am just narrating right now? The chromatin and many other terms that I'm going to speak about, such as the histones and all of these terms, are like the wrapping of the wrapping, the protection that covers the DNA and that helps to give shape to the chromosomes that are having all of the sequences and all of the genes of our cells. So let's picture that you are going to give a present. The present has a wrapping. So it can have a wrapping in a box. It can have a wrapping the box, and then to cover the box, you are going to use paper and you are going to use a ribbon or many ribbons. So all of the cover of the box that is going to have the present that you are going to give to the person is the chromatin and all of these factors. So all of the wrapping is part of the chromatin. The ribbons can be, if you imagine ribbons in the wrapping of this present, those are called histones, and all of this is the protection of the genes. But all of this protection of the genes is also extremely important in terms of the expression and the regulation of the expression. How? If I have a wrapping in a present that is difficult to take it out, I will need help. For instance, if I am a child, or even if I am an adult, sometimes we have problems to open a present because the wrapping is extremely well covered or is tight, and it has a lot of ribbons, it has a lot of paper wrapping or whatever it is, and you need to take out all of the layers in order to get to the present, which is going to be your sequence, your gene. So in terms of a cell, you also need to take many of these factors and to start unwrapping and unfolding many of these proteins that are stimulate the wrapping so that you can get to read the sequence. And all of these elements that are going to protect and cover the genes are subject to being regulated by epigenetic marks. The epigenetic marks can be very different. Can be acetylation, which is a chemical group. Methylation is another chemical group. But all of these groups are going to attach to this kind of wrapping, and it's going to be more difficult to get it out. Let's imagine that it has a lot of scotch tape on this wrapping of the present. And in order to break it, you need help, or you need scissors in order to cut it off. Well, the same happens with your genes. In order to remove these kind of epigenetic marks, you need to have a healthy lifestyle. And if you have a toxic lifestyle, you are going to start adding these kind of protections and stains and things that are going to complicate the reading of your genes. If you complicate the reading of the genes, the proteins that you are going to have are going to be extremely low quality. And that's what is going to lead to symptoms, and the symptoms are going to lead to a disease. So that's the whole process summarized. I'm going to go deeper, but this is how it works. So to mean that the inheritance of alternative chromatin states in the absence of changes in the DNA sequence, we have DNA methylation that was proposed early as a carrier of epigenetic information, with subsequent work revealing that chromatin proteins and non-coding RNAs are also important for this process. So all of these proteins, the chromatin proteins and the non-coding RNAs, are going to complicate the reading, and they have epigenetic information because they are going to protect, either turn on or turn off certain kind of sequences, genes, and are going to give access or complicate the access to the reading of the sequence. For example, histone variants and histone modifications can influence local chromatin structure. So this is I'm just talking about the wrapping paper and all of these things that are going to protect the genes, either directly or indirectly. Why? Because it is just the cover, the protection of the gene. That's why it can be direct, because it can completely cover and not allow the other transcription factors to position themselves and to give signal and to have a very good signal post for the enzymes to be positioned here and reading the sequence. That's why all of these modifications can be heritable, but they can be reversible, and they are governed by a series of writers that deposit them, and readers to interpret them, and erasers to remove them. So it's like I explained, if I want to publish this book in Spanish, I will need a translator, I will need an editor, I will need a company to publish it, etc. I will need a lot of members or people that are going to help me to publish the book in the language. So all of these helpers are the ones that are going to also participate in our genes, and they are the writers, they are the readers, and they are the erasers of these epigenetic marks. Finally, the high order of 3D chromosome folding, so that is all the wrapping of the present, which is your gene, is also thought to modulate the expression and might contribute to inheritance. Of course that it is going to contribute, because if you don't have access, if you don't have a clear signal of where do I have to read, or what kind of sequence do I have to read, I'm not going to have the best quality of the protein. That's what is going to happen. Since, since 1942, when the word was first coined, epigenetics has been redefined multiple times. In this review, in this article, the term epigenetics is used to mean the study of molecules and mechanisms that can perpetuate alternative gene activity states in the context of the same DNA sequence. So we have one sequence. Let's picture that it is my hand. Just this is the sequence. The sequence can have alternative gene expression and alternative activity states depending on the epigenetic marks. So depending also of the protection that I have to remove and the kind of marks that I am producing, either acetylation or methylation, or other types like the RNAs that are not codifying, the non-codifying RNAs, and all of these mechanisms and names that I'm going to mention. So all of these players are going to impede the reading or facilitate the reading. This operational definition has several implications. First, it encompasses transgenerational inheritance, as well as mitotic inheritance and the persistence of gene activity or chromatin states through extended periods of time, even without self-division. For instance, in long-lived post-mitotic cells, such as adult neurons. So these type of cells, neurons, don't suffer meiotic division. So they don't give rise to germinal cells. They just have a longer period to renovate themselves and to suffer this division of cells. But some of them don't divide at all because they lack many of the machinery to be divided. But we can form from germinal cells other neurons, and we produce neurogenesis. And in these kind of cells, we have a different kind of protection in terms of the wrapping. If we have a very tight or very obstructive kind of wrapping to the reading of the sequence that is going to give rise to the neurons, we are impeding the quality of neurons and accessory proteins that are going to give rise to the neural pathways. In mitotic inheritance, mitotic means that are the cells that are constantly dividing, such as any other cell of your body is constantly dividing and renewing. One should consider that the genomic sequence of these individual cells, whereas in transgenerational inheritance, one should consider the DNA of the whole organism, which includes its microbiota, and that microbiota can contribute to the inheritance. Imagine this paragraph that I just started to discuss and to reflect on. Just the mitotic inheritance that I have, all of the cells of my body are constantly renewing. That means that I can change and I can reverse the epigenetics of many of these cells. But there are some cells, such as the neurons, that are not going to have this kind of multiple division cycles. So if I don't have this kind of division cycles in many of the neurons, many of that information is going to pass like that to the next generations, which can be even 7 generations. Yes, you heard me correctly. All of the information, all of the genetic information can pass up to 7 generations. So just think about that. You are inheriting all of that information to 7 generations of your family. So if you have a clean, healthy lifestyle, you are going to give them the best information and they are going to have a much better outcome in terms of the health and in terms of expressing certain kind of diseases. But if you give a very poor inheritance in terms of all the information that you have and many of these epigenetic marks, you are going to predispose them. Not is not a sentence, is not the destiny, but you are going to predispose them to certain kind of diseases. So that's the power and that's the message of epigenetics. And the microbiota is included there. What is the microbiota? I have already talked about that, but I'm going to define it again. It's all the microorganisms, which include bacteria, fungus, and viruses inside of your intestinal flora, and they are also present in the mucous membranes of your nose, of the ears, of your mouth. So all of these bacteria, fungus, or viruses that we have are going to give rise to many of the proteins that we need and many of the neurotransmitters that we need. They are our helpers as well. Well, this definition of epigenetics that I just mentioned explicitly extends the usage of epigenetic to regulatory processes because we are constantly regulating this kind of information. If we start having a much healthier lifestyle, we are going to clean many of these epigenetic marks and the access to the information is going to be easier. The reading is going to be much better. And of course, the interpretation and expression is going to be high quality. If not, we are leading ourselves to failure and to have a very poor quality of proteins. So all of these regulatory processes involve molecules known to participate in epigenetic inheritance, even when not addressing the epigenetic memory function per se. Why? Because the memory of your epigenetics is all the information that you already have. But if you change your lifestyle, you change the way the information is going to be expressed. That's the most important message. An appreciation of the role of chromatin as a carrier of epigenetic information that can propagate active and silent activity states during cell division came from the study of different biological processes and model organisms. These include, to name a few, heterochromatin inheritance, genomic imprinting, the parent of origin-specific repression of certain genes in mammals, and all of these kind of processes and all of these kind of factors, these complexes are thought to perpetuate the functional responses by modifying the histone proteins in the chromatin and by binding their own histone marks in order to convey a stable inheritance. What is the meaning of all these scientific jargon and terms? To understand better, let me take another example of this box, and let's suppose that this box inside has a present, and I have a wrapping, and I have ribbons, and I have many things that are going to be protecting the present that I want to give. So all of the wrapping, all of the ribbons, everything that I am having in the outside, let's say, are the chromatin, are the histones, and are many of these factors that are going to interfere with the reading of the present that is inside. And many of these can have epigenetic marks, and the epigenetic marks are going to regulate how much are you going to see, how what kind of fidelity and assertiveness you are going to have in terms of the position of the enzymes in the correct sites in order to read the sequence that is going to give you the best expression of the protein. That's what it is going to happen. So all of these wrapping and ribbons and everything that is protecting is also involved in unstable inheritance or an unstable and toxic inheritance. Depends on you. Depends on the kind of epigenetic marks that are going to inherit to 7 generations. So let's go now to the history and the definitions that have been throughout the years of epigenetics so that you can see how much we are advancing in this kind of information and how much we can control of the process right now. So in 1942, Waddington said that epigenetics is the processes by which the genotype brings the phenotype into being or into expression. So it was a very basic information and definition. He just says it's all of the processes that are going to give, this is the genotype, this is the sequence, this is the genotype, the information that is going to give me the shape, the color of something, whatever it is, a protein or a whole individual or a whole function of the cell. Now, in 1958, another author or researcher named Nani, he said the systems that regulate the expression of the library of specificities, that is, the genetic material, which is meant to be the DNA or RNA sequence. So now it is more specific. It is all the regulation of expression. Now we know that there is a regulation. Now we know that there are several systems, some of them in the sequence, some of them outside in the wrapping, and this is controlling the DNA or the RNA. Now we know of another sequence. The RNA sequence is one of the interpretations of the DNA sequence because in order to obtain the RNA, we need the DNA. Now, in 1975, Riggs has another definition, which is also going to be shaped in 1996 by other researchers named Holliday, Martinson, and Russo. So they say mitotically and or meiotically heritable changes in gene function that cannot be explained by changes in DNA sequence. So now they are saying that all of these changes are going to happen in the multiple division processes, which they can, but they can also be previously labeled or previously positioned, previously generated before the cell divides. And they change the gene function. That's what they know. And they cannot be explained by changes in DNA sequence because they didn't see changes in the DNA sequence. They saw that they have some stains, some labels, such as the methylation or acetylation or other types of chemical tags, let's say, but they didn't know what they were going to do or what it was going to give rise to. And what they knew already is that the DNA sequence wasn't changing. It was just being regulated. Bird in 2007 says epigenetics is the structural adaptations of chromosomal regions so as to register, signal, and perpetuate alter activity states. So now we know that there is an structural adaptation of regions in the wrapping that are going to register signal, so they are going to send a message, and perpetuate, they are going to be inherited alter activity states. So they are going to alter the expression of the genes. In 2017, Greeley and Lapalane said epigenetics is the properties of a cell mediated by genomic regulators that confer on the cell the ability to remember a past event. This is more mystical now. We are involving past lives, let's say, or past processes from other people that gave us the information. So our parents, grandparents, great-grandparents, and etc. 7 generations behind. So all of these properties of a cell are mediated by genomic regulators, the regulators of the access of the information, and they are going to confer an ability to remember how well it is a past event. So how well it is the protein. But that depends on you, again, on the lifestyle that you have. Nikoglu in 2017 said, they are various processes, various intracellular factors that have an effect on stability of developmental processes through their action on genome potentialities. So depending on the potential of the genome of the sequence, you will have either stability or instability, depending of the development processes that you have followed. And they are various, they are a lot of intracellular factors. So it is giving us all the responsibility to us to change our genes, to turn them on, the ones that we want to produce with high quality proteins, and to turn off the ones that are toxic, and to eliminate many of the genetic factors, epigenetic factors, or epigenetic marks. Which are the players? Let's just go quickly into who are some of the players. Some of the players of these whole epigenetic processes are called heterochromatin components. What are these components? They are pericentric heterochromatin containing a large number of proteins, but the most distinctive feature of them is the presence of megabase size repetitive DNA domains that are coded in histones. So they have a lot of wrapping. These are part of the wrapping of the present of your gene. The polycomb proteins, they are early genetic sequences classified as polycomb. That's the name, that's the technical name. and they are abbreviated as capital P, C, G, and they are three thorax, that's the name from where they found that, into two antagonistic groups. Some of them maintain the memory of spatial patterns of expression of homeotic genes throughout development, and some of them are epigenetic regulators of transcription. So they give rise to the identity of the cell. They affect the differentiation of the cell, and either they lead it to a very high quality protein or they predispose you to a disease. That depends on you again. Now, non-coding RNAs. Non-coding RNAs, which are abbreviated as ncRNA, belong to several classes, and neither their production nor their function can be generalized. Many of these, such as the microRNAs, regulate post-transcriptional processes, whereas others are involved in transcriptional regulation. This is extremely important. Why? Because one of the new technologies that we are seeing now in certain medications, that you know what medications I'm talking about, are made now of RNA. So we are saying here, we don't know what can happen in the regulation post-transcriptional processes. So we don't know what these kind of sequences can do and how they can regulate the expression of certain kind of sequences. And they are also involved in transcriptional regulation. So they can lead to a bad expression of any kind of protein, which is going to lead to a disease. So just pay attention to that. Now, the other ones are DNA methylation, which is the most common. And these are the mechanisms that allow DNA methylation to be copied during DNA replication, represent one of the best understood epigenetic systems, and they involve specific proteins that recognize sequences or islands of cytosines and guanine, which are two of the most common in this kind of sequences, nitrogen base in your DNA. And they can be hemimethylated or they can have high methylation. And those kind of methylation percentage, let's say of the sequences, are going to interfere with the expression of the genes. Now, what are the genetic effects on epigenetics? The genome of each individual experiences both natural and environmentally induced mutations. So we constantly are having mutations that can be beneficial or can lead to certain kind of diseases, depends on us. And some of them, many of them, are environmentally induced. While most mutations are neutral, sequence polymorphisms can affect epigenomic landscapes. What is the meaning of this? This means that if I have one mutation, two mutations, three, might not lead to anything. It is just neutral. But if I have 10, 100, 200, or 500, or 1,000 mutations, which are going to be polymorphisms now, then I will affect the epigenomic landscape. So what is going to give rise to a defective protein, a low quality protein? Now, Chromatin and DNA methylation in mutagenesis. How do they interfere? The chromatin, remember that is the wrapping, and DNA methylation are the chemical groups that are going to attach to the sequence of the DNA, to certain sequences of the DNA. Mutation rates vary in different parts of the genome at different stages of the life cycle, and in diseases such as cancer, where they depend on the cells of origin, environmental exposure, and the cancer type. Mutation rates can be affected by DNA methylation and nucleosome positioning. This nucleosome is another part of the wrapping that I have to protect the sequences. Higher order chromosome folding also influences mutagenicity. So I need to protect the wrapping and I need to protect the sequence from these epigenetic marks. Now, what is happening in the epigenetics and disease? Changes in the level of DNA methylation, histone modification, and changes in non-coding RNAs functions are common in disease, as are mutation in epigenetic components, which is the wrapping. The ability to distinguish driver from passenger roles for epigenetic alterations will make it possible to identify diseases in which epigenetics might affect the diagnosis, prognosis, and therapy. So what we are trying to see now in the current time of these kind of epigenetic modifications is which ones are the most dangerous and how can we clean them? What type of environmental activities are the best for each type of person? This is the personalized medicine that we are going to arrive there in a very few years. Dissecting the interplay between epigenetic components and other disease pathways will also allow the development of combinatorial intervention approaches. So that's the science that we should be experiencing more. And that's a science that has to be deeper now, because the more we know how this epigenetic landscape behaves and who is the driver and who is the passenger, so who has more responsibility, what type of modification have more responsibility in the expression of the gene, in the production of a low quality protein, and then a disease, that's where we can have more therapy and more recommendations of changing certain kind of lifestyle. Now, what is happening with epigenetics of aging? The application of the machine learning and the high throughput DNA methylation data has identified indicators of chronological or biological age. One study found that changes in the CpG sequences methylation, which are the cytosine and guanine islands of being methylated, being tainted or damaged at 353 genomic sites, produce a score that was highly correlated with age across tissues. So this is one of the most important breakthroughs in order to discover that we have an epigenetic clock. Strikingly, a comparison of different molecular predictors of age indicated that the epigenetic clock is the most highly correlated to biological age. So we have to take care of this biological age. It is not the same of the chronological age. You can be 50 years of age chronologically, but have a biological age of a 40-year person or 35-year person. That depends on your lifestyle. For instance, in my case, I have 15 years less in age, in biological age of my cells, because I have um, a scale that measures the biological age. So when I weight on that scale, I measure those kind of factors, and I know that I have reversed my age or maintained my age in a younger state just due to the lifestyle that I have, which is, of course, the nutrition, the emotions, meditation, and, of course, the kind of exercise that I'm doing. So, all of these factors are contributing to the expression of your genes. So, we have to open our eyes to the responsibility that we have and to how we can reverse the age. Furthermore, epigenetic age is adversely affected, and they found out in these kind of studies, and is accelerated by a high body mass index, so obesity or overweight, starting with overweight and leading with obesity. Whereas it is reduced by high levels of education or physical activity. Just picture this, the level of education that you have, and it doesn't have to be in terms of the degrees that you have, has to be in the kind of information that you consume, in the kind of activity that you have in terms of your brain, and the kind of habits that you have in order to challenge your brain to learn something new. A low body mass index and consumption of fish, poultry, and fruits and vegetables are very good in terms of affecting your epigenetic age for the reversing of the age. Many of these 353 sequences investigated are located close to poise promoters or bivalent genes, or to active promoters. What are the promoters? The promoters are sequences that are going to enhance the reading. They are going to give a much better signaling mechanism for the reading, and they are going to help in the translation process of how a protein is going to be produced. So this suggests that aging may correlate with reduced plasticity in the expression of these kind of bivalent genes, which might resolve into repressed or active states, and with active genes changing their expression levels. So all of this is giving us all of the hints, all of the information, which is the integration of clinical measure that can affect the phenotype age, and how all of these sequences and epigenetic marks are better predictors of the lifespan of the health of a person. So we are now establishing mechanistic links between the aging process and the variation in the CpG methylation, and they will be critical in order to identify the causes of aging. This is the future that we are now arriving. The future is studying what type of sequences, what type of methylation, what type of processes in the graphene or in the sequence, which are more important and which are leading to aging of the cells, reversing the age of the cells, and producing a low quality that is going to lead to a disease. That is the most important thing of epigenetics. We can reverse diseases, we can produce diseases, and we can even change all of the landscape of the epigenetic genome, so the information that we have within. It is on you. The the responsibility, the driver's seat is your consciousness, are your beliefs, are your habits, are your behaviors. All of these is going to lead to a healthy expression of your health or a disease. It's on you. Now let's see what is happening in terms of the advancement of epigenetics and how can we have an intervention in the way we regulate our genes. And we are going to also see how it is leading to one of the most threatening disease, which is, of course, cancer. So let me just show you what it can do in terms of having an expression on your genes. Let me show you what is happening and what is the science and how it is advancing in terms of this roadmap. Let's see what is happening here. Roadmap of the DNA methylation phenol age. So how the developing of a multi-system estimates the phenotypic age based on clinical markers and validate associations with all-cause mortality, cause-specific mortality, coexisting disease count, and physical functioning. So this roadmap depicts our analytical procedures and they develop an estimate of the phenotypic age based on clinical measure. So now we are going to use, well, researchers are using and they are training a composite epigenetic predictor of phenotypic age called DNA methylation phenol age and validate the associations with all-cause mortality, coronary heart disease incidence, coexisting disease count, physical functioning, disease-free status, age at menopause, cancer incidence of lung and breast cancer, familial longevity, mild cognitive impairment, Alzheimer's disease neuropathology, Parkinson's disease prevalence, Down syndrome, HIV positive status, chronological age in 35 cells and tissues that they are going to choose and define, which are the most important in terms of the chronological age, socioeconomic status, race, ethnicity, diet, physical activity, metabolic syndrome, smoking habits, and obesity. All of these factors are going to be merged, analyzed, and they are going to give us a lot of information in terms of what can happen with your life. Identify an underlying biology of the DNA methylation phenol age in the sequences of cytosines and guanines and the composite score. So how does this methylation is going to lead you either to clean your genome or to taint it in terms of leading to a disease? They are going to give you these kind of scores, GO enrichment, pathway enrichment, LTL correlation, differential gene expression, CpG island enrichment, heritability analysis, immune cell associations, CpG overlap with existing clocks, loci-specific DNA methylation versus the transcription. This is in terms of the specific sites of where is the methylation of your DNA, polycomb group protein targets, so which kind of proteins in the wrapping are interfering with the expression of your genes, chromosomal location, and longitudinal change. All of this map is using multiple cohorts, aging-related outcomes, and tissue and cells. They examine the underlying biology of the 513 CpG and the composite DNA methylation phenol age measure. Using a variety of complementary data, gene expression, blood cell counts, and various genome annotations tools, including chromatin state analysis and gene ontology enrichment. This gene ontology means the origin of the enrichment, so how good is your inheritance in terms of the gene sequences that you are inheriting. And now what is happening in terms of the diseases? How it is impacting the methylation in terms of age and in terms of the alteration in cancer. So as you can see here, DNA methylation-based age, as we increase this methylation in certain specific cells that control this chronological epigenetic clock, they are going to lead to a more extensive or more advanced chronological age. The less or the more we clean these epigenetic marks in these specific cells, we are going to control the biological age of our cells. Now, in terms of cancer, tumorigenic insults, look at this word. You are insulting your body with the type of activities, behaviors, or habits that you are having, because you are leading your sequences to be tainted, to be marked with these epigenetic chemical groups that are going to interfere with the reading and the quality of your proteins. Which are the tumorigenic insults? Smoke, pollutants, chemotoxic agents, aging, nutrition, of course, exercise, meditation, etc. Many of those, if you don't have exercise, if you don't have meditation, if you don't regulate, if you don't do anything, all of that lifestyle of stress is going to lead for these tumorigenic insults. Mutations in epigenetic regulators, polycomb, 3-thorax, DNA methylation, chromatin, and remodelers. Many of these are the wrapping changes, and they are going to have altered cell signaling, epigenetic amplification of altered cell states, maintenance of altered cell states by transcription factors dependent on regulatory feedback loops, and they are going to lead to cellular effects, loss of cell cycle control, which leads to overproduction and of these kind of cells, senescent cells that are no longer productive, let's say. Loss of differentiation, loss of cell addition, senescence bypass, so they don't know when a cell has to die or not, and many of these cells can lead to tumors and cancer. Increase in proliferation, which also leads to cancer, increase in cell-cell heterogeneity or heterogeneity. So that means also cells that don't know the function, they are altered, they don't know what to do, and they start leading to these tumorigenic cells. What is doing the epigenetic clock? Or the epigenetic clock consists of a specific set of genome CpG sites whose levels of DNA methylation change progressively with age, leading to an estimate of age based on DNA methylation that correlates tightly with chronological age. Rather than a global change in the methylation levels, some of these age-related CpG sites show increased methylation, which are these black marks that are here, black lollipops, and the red outline, and some of this, whereas others show decreased methylation, as we can see here, the white ones. So it depends on us if we decrease the methylation or we increase the methylation, and we lead to more aging of our cells. In terms of the disease, the relationships between changes in DNA methylation and chromatin architecture in aging remains to be investigated, as well as the cause-consequence relationships between aging, DNA methylation, and gene expression changes. We are still knowing more about the kind of changes that we can produce, but we know already that it leads to more aging. The genes encoding epigenetic components such as DNA methylases and demethylases, polycomb 3-thorax chromatin remodelers, DNA methylation component, and all these CTCF sequences are frequently mutated or dysregulated in cancer. So we don't know what is happening, but we know what is interfering in the expression of healthy cells and how many of these tumorigenic insults are leading to disease. And DNA methylation components frequently mutated or dysregulated are present in cancer, and they are often a result in environmental insults or physiological changes such as aging. That's what we know already. That's what you can do to reverse. We know that many of these is reversible, and many of these are being shown here in the white marks. And the more you promote a healthy lifestyle, the more you add an integral lifestyle with these 5 pillars of health that I am constantly teaching, that's the way that you are going to clean your genes. And that's the way you can even reverse many types of cancers and many types of chronic diseases. So thank you for paying attention also to this final presentation that I'm giving. And this is all the information that you can have in terms of the epigenetics and the most recent published articles in terms of how it is advancing in very fast pace. So let's do something to contribute to these kind of results, and then let's start to reverse our age. Thank you and have a wonderful day. Bye.